The theory of population genetics is a fundamental concept in evolutionary biology that applies the principles of genetics to study how the genetic composition of populations changes over time. It provides a mathematical framework to understand natural selection, genetic drift, mutation, gene flow, and recombination, explaining how species evolve and adapt to their environments. By combining Mendelian genetics with Darwinian evolution, population genetics bridges the gap between microevolution, small genetic changes within populations, and macroevolution, the formation of new species over time. Population genetics focuses on allele frequencies, the proportion of different genetic variants, alleles, in a population, and how these frequencies fluctuate due to evolutionary forces. The study of these changes allows scientists to predict how populations evolve, understand genetic diversity, and explore the genetic basis of adaptation and speciation. Key principles of population genetics: Hardy-Weinberg equilibrium, a fundamental concept stating that allele frequencies in a population remain constant from generation to generation unless evolutionary forces act upon them. This principle serves as a baseline to detect evolutionary changes and is represented by the equation p2 plus 2pq plus q squared equals 1, where p and q represent the allele frequencies. Deviations from Hardy-Weinberg equilibrium indicate that evolutionary processes are occurring. Natural selection, a driving force of evolution where individuals with advantageous genetic traits have higher survival and reproductive success. Over time, beneficial alleles increase in frequency, leading to adaptive evolution. Genetic drift, a random change in allele frequencies due to chance events, particularly in small populations. This can lead to loss of genetic variation, fixation of alleles, and even speciation. The founder effect and bottleneck effect are examples of genetic drift that significantly impact population genetics. Mutation. The ultimate source of genetic variation, mutations introduce new alleles into a population. While most mutations are neutral or harmful, some provide advantages that natural selection may favor. Gene flow, migration. The movement of alleles between populations through migration influences genetic diversity. High gene flow can prevent populations from diverging, while limited gene flow can lead to genetic differentiation and potential speciation. Recombination. The reshuffling of genetic material during meiosis increases genetic diversity by creating new allele combinations. Recombination plays a crucial role in the evolution of sexually reproducing organisms. Applications of population genetics. Conservation biology helps in assessing genetic diversity, inbreeding risks, and designing conservation strategies for endangered species. Medical genetics, used to understand genetic diseases, human evolution, and how populations respond to environmental changes. Agriculture assists in selective breeding programs to enhance crop and livestock traits. Forensics and ancestry studies helps trace human migration patterns and analyze genetic ancestry through population studies. The theory of population genetics remains a cornerstone of evolutionary biology, genetics, and ecology, providing insights into how life on Earth continues to evolve. With advances in genomics and bioinformatics, researchers are now able to study genetic variation at an unprecedented scale, further unraveling the complexities of evolution and adaptation.